gonna take a step back from biotechnology for a few minutes. Now, instead of talking about how genetic modification of biological systems for human benefit, I just actually want to take a step back and talk about plants and how they interact with their environments. And also talk about specifically how plants interact with biotic stresses and bacteria. Because actually, when it comes to the next method for getting DNA into plants, humans didn't actually invent that method. A bacteria called Agrobacterium tumefaciens did. We will not ask you how to spell that on an exam. So this is a bacteria that is found normally growing in the soil. So here's a bacteria growing in the soil. We also have plants. Here's our plant. And I'm going to abbreviate this as agro. So because plants can't get up and walk away, they have to interact with the things in their environment. But one of the problems with that, of course, is that there's all sorts of bacteria around. So sometimes plants do get wounded. What happens when they get wounded is they give off a signal. So if you remember from that campus walk we went on, we talked about some of the signals that plants produce, some of the chemical compounds they make. So when plants do get wounded, so here's our wound now, they send out a signal. It's a phenolic compound, so here's our plant sending out a signal. And this agrobacterium involved a way to actually recognize that signal. So we have our wounded plant, agrobacterium recognizes that signal. Agrobacterium is a pathogen of plants, so when it does perceive that signal, it is able to infect the plants. And then what's really interesting about this infection is that the plant, after it's been infected, begins to form a large tumor called a crown gall. So this is not typical of what you would normally think of when you're talking about a bacterial infection, right? So normally you'd think bacteria would go in, probably kill the plant. But in this case, instead of killing the plant, this interaction induces the formation of a tumor. I'm going to show you what, let's see. Here's what some of these tumors on plants look like. So I know this one has less of an impact here, but this is a tree outside of one of the big classroom buildings on the UC Davis campus. And you can see this whole tumor here is absolutely enormous. This tree is enormous. So this isn't an interaction that kills the plant. Although it's certainly a parasitic interaction, there's a one-way transfer. Plant gives the bacteria nutrients, bacteria only takes and gives nothing in return. This is certainly not a good thing for the plant. But this is an interaction that is not a death sentence to the plant. So it's sort of weird, right, that instead of what you would normally think of with a plant bacteria interaction or any sort of bacterial infection, that we have this tumor forming. So let's see. So some scientists took a closer look at the cells of this tumor. So here's our plant cell. and the chromosomes of the plant cell in the nucleus, and what they found was integrated into the genome of the plants is some of the DNA from that agrobacterium. So these agrobacterium have figured out a way to get some of their DNA out of their cells, into plant cells, into the plant nucleus, and into the genome of the plants. So a little bit crazy to think about. So actually, sort of this idea of genetic modification being unnatural, this agrobacterium actually invented it first. An oddly sort of natural process. It's sort of crazy to think about that this bacteria, through sort of, that random mutations in evolution, acquired the ability to not only sense the plant compounds, but to transfer its DNA into the plant. So, a little movie to show you of this wrong way, happening. Then we'll talk about what's actually being transferred, but I want to show you this movie